Hey students, it's Mrs. Owens. Let's go over pages 12 and 13 of your note packet. So pages 12 starts out talking about the cell membrane. You may have heard of the cell membrane, you may not be familiar with that word, but the membrane is basically just the outer surface of the cell. It's what keeps everything contained inside the cell and keeps everything outside the cell out. So let's start by reading this first paragraph on page 12. So our first paragraph says, all cells contain cell membranes, which are composed of lipid bilayers. If you think back to our first unit, we talked about lipids a lot, right? Lipids are fats. So your cells, the surface of them, are mostly composed of fats or lipids. We also sometimes call them phospholipids. And so sometimes we call it a phospholipid bilayer. The lipid bilayer gives cell membranes a flexible structure that forms a strong barrier between the cell and its surroundings. The cell membrane regulates what enters and leaves the cell and also protects and supports the cell. These lipids that make up the cell membrane are composed of a hydrophobic tail and a hydrophilic head. So for that first paragraph, just make sure to underline those two sentences. The first one being the cell membrane is composed of lipid bilayers. And then second of all, make sure to do this last sentence here. Oh, sorry, that was crazy. I think that will go away. Yeah, these lipids that make up the cell membrane are composed of a hydrophobic tail and a hydrophilic head. So let's go ahead and write the definitions in the space below. So you guys probably already have some knowledge of what hydro means and what phobic means or what philic means. If something is hydrophobic, if you have like a phobia, you're scared, right? And so this particular term, it means that the tail of the lipid molecule is scared of water. It's kind of a silly term, but yeah, we say the tail is hydrophobic, it's scared of water, and then we say the head is hydrophilic, which means it loves water, which is kind of unique, right? That we have this molecule that both loves to interact with water, part of it does, and then the other part doesn't. So let's go to this space below. In your directions, it says draw the shape of a lipid molecule and label the hydrophobic tail and the hydrophilic head. Our lipid molecule kind of looks like a tadpole. So the circle here at the top, that's going to be the head. And then it's got these squiggly tails. In this particular molecule, I drew two tails, but sometimes our lipid molecules can have three. Sometimes they can have straight tails, and so there's a lot of variation. And of course, these are molecules, right? So they're carbon, oxygen, hydrogen, all of those elements form these really cool structures. And we're not going to look specifically about how they bond and stuff like that. I just want you to know what the general shape of this molecule looks like. And then go ahead and label the head hydrophilic and the tail hydrophobic. All right, let's go to the next part here. It says if you were to submerge a bunch of phospholipids in water, how would they arrange themselves? Draw the arrangement in the space below using 10 lipid molecules. There are two different options here of what could happen. So let's say we throw a bunch of lipids into some water. If they weren't completely submerged underneath the surface, it's most likely that our lipids would arrange themselves as option 1 here. So in option 1, we've got the heads underneath the surface of the water with the tails sticking out. And that's totally fine, right? Because the tails don't like the water, so they're sticking out of the water. And then the heads are underneath the water because they like to interact with these water molecules, right? I drew the water molecules as blue oxygens and red hydrogens. They look like the little Mickey Mouses. Okay, but let's say we were to push all of these molecules underneath the surface of the water. How would they arrange themselves? And most likely they would arrange themselves as option 2 here. So in option 2, we've got a bunch of heads that like to interact with the water. And then the tails, which are hydrophobic, kind of point inward so that they're not touching any of the water molecules. So it almost kind of looks like a bubble, right? It creates this little bubble where the heads are interacting with water, but the tails are not. So yeah, this is page 2. This is our introduction to phospholipids or lipid molecules that make up our cell membrane. So now that we kind of understand the nature of these molecules, we're going to talk about how they arrange themselves in our cells. So let's go ahead and flip to page 13. If you need more time, feel free to pause the video here too. So let me go ahead and pull up page 13. I'll make this nice and big. All right. So in 1925, there was this famous experiment that was performed by two Dutch scientists. One was named Gorder and the other Grendel. They extracted membranes from red blood cells and they analyzed them. They found that there were twice as many lipid molecules compared to the original surface area of the red blood cells. So go ahead and under that last line, they found that there were twice as many lipid molecules compared to the original surface of the red blood cells. So why are Gorder and Grendel's findings surprising? Well, we would expect, right, we know that there are lipids that make up the membrane, but originally we would expect that the ratio of the lipids to the surface area of the red blood cell would be just a 1-to-1 ratio. But that's not what Gorder and Grendel found, right? They found there were twice as many lipid molecules, so it's more like a 2-to-1 ratio. So this experiment here was really important in understanding the structure of our cell membranes. So let's go ahead and redo our diagram, right? On page 12, we kind of made that bubble with all the tails facing inward. But how do lipids really arrange themselves in cells? So we have to consider the two facts down here below. Our first fact is we know that Gorder and Grendel's experiments, according to their experiments, that they had twice as many phospholipids per cell than we originally thought. Okay, so keep that in mind. There's twice as many phospholipids as we originally thought. And number two, the center of the cell is also full of water. I already drew the picture here, so I probably gave it away. But this is the model that Gorder and Grendel came up with. So they said there's water on the outside of the cell, right? We can see these water molecules on the outside. We can see these water molecules on the inside of the cell as well. And phospholipids, or lipids, arrange themselves so that they've got heads on the outside, they've got heads on the inside, and their tails face inward. So the tails are always facing in, or like facing towards each other. So in your model, you can draw what we call this bilayer, bi meaning two, this two or double layer of lipids that make up the membrane of the cell. Yeah, and you don't have to draw the water molecules in your picture, but make sure to draw this phospholipid bilayer, or this lipid bilayer. It's going to take you a second, but when you are done, you can go ahead and move on to this last paragraph here. And it says some molecules are able to squeeze through the lipid bilayer into the cell. So for example, our cells need water, and they need other things that go through the cell, right? But for these other molecules, they're not able to go through the lipid bilayer. So they have to have some sort of means or door or tunnel to get in and out of the cell. So scattered throughout our cell membrane, we've got a bunch of proteins that act as channels or tunnels. So in your diagram, you can just draw 3 different proteins. Your proteins are going to be completely embedded in the membrane. One side of the protein is going to face outward, outside the cell, and the other side of the membrane is going to face in the cell, right? So that these molecules can pass through this lipid bilayer. So this is kind of how we represent it. I just drew my proteins as blobs or squares or circles. But let me show you another model that represents things moving in and out of the cell. I'm going to Google cell membrane. And I really like this diagram here, so let's look at this. Keep in mind, everything is 3D, right? Cells are 3D. They're spheres, they're not just circles. Our cell membrane is 3D, and so are our proteins here. So if we look at this model, our pink circles represent the heads of the lipids. The green squiggly lines are the tails. And then these giant purple shapes are the proteins that do different jobs. Most of the proteins that we're going to talk about are these channel proteins, and they kind of look like a slide for molecules to come in and out of the cell. And then if you look at this little ring over here, it actually it looks like a little circle or a little hole that things can go in. So keep in mind this is 3D, right? And these proteins act as doors. When you are done with your notes, you are able to finish the Fainting Goats assignment. So make sure to do that on Canvas. And if you have any questions, you can send me an email or come in after school, and I'm happy to help you with those assignments. But thank you for watching the video, and I hope you guys have a good day.